Our last speaker before the coffee break is Dr. Linda Ercoli. She's Associate Professor of the University of California, Los Angeles, and Director of the Geriatric Psychology Service in the Semel Institute. And she's currently investigator of the Alzheimer's Disease Research Foundation, she's going to present us some work on imaging brain amyloid and tau levels in adults with Down syndrome using PET studies. Thank you, Linda.
Okay, thank you, everyone. Thank you very much for inviting me today to speak. My name is Linda Ercoli. I'm from UCLA, and I'm here to talk about some of the latest neuroimaging work that we're doing at UCLA. And I'm going to represent the work of our laboratory that's under the direction of Dr. Gary Semel. And we're going to talk about imaging the brain of amyloid and plaque in the brain of and tau in adults with Down syndrome using positron emission tomography. But first I'm going to give a background on some of the work that we've done in Alzheimer's disease, and then talk specifically about some work that we're doing in Down syndrome. So the field of study of plaque and tangle imaging definitely has strengths and weaknesses. Some of the strengths is that the probe that I'm going to talk about today that is used to image plaques and tangles is called FDDNP, and it's a PET technique that Dr. Simmons had talked about PET techniques previously. And so this images amyloid plaques and neurofibrillary tangles in the brain, and it correlates with age and genetic risk for Alzheimer's disease. But the one weakness is that the studies are very limited using this probe in Down syndrome. And there are many gaps to be filled that we'll talk about as the talk progresses. But some of the gaps is, can this really detect dementia? Because this is one of the challenges with Down syndrome. Is it going to be helpful in detecting dementia and predicting outcomes and status in Down syndrome? Can we use it to monitor treatment response as maybe anti-amyloid or anti-tau treatments are being developed? And can we use this in Down syndrome to study the relationships between FDDNP binding in the brain and any genetic markers? And finally, I think this is playing a devil's advocate, but in Down syndrome, what are going to be the utility of senile or amyloid plaques and neurofibrillary tangles for the early detection of AD in people with Down syndrome? And definitely the resources that are needed from a neuroimaging perspective, more biomarkers and probes. Recruitment is very difficult, as we're finding out. Funding is limited, and lobbying private foundations and government agencies is going to be important. And in one of our previous talks, we talked about how important it is to not only study Down syndrome for the individuals with Down syndrome, but also because of the relevance of Down syndrome to other degenerations that affect millions of people. And we can return back to this, but how the community can help create this is going to be starting with what we're doing today, getting minds together, perhaps establishing consortiums or data banks. One thing that I've noticed, especially because I'm a neuropsychologist by my training, is that everybody uses different instruments, and it's very difficult. You have lots of people being studied, but using different methodologies. Is there any way we can have a core set of variables that, even if we just have a few, will help essentially expand our subject pool and also assist with facilitating collaboration? And the idea of this, too, is to actually enhance the lives of people with Down syndrome. So improving identification of Down syndrome individuals that may be developing Alzheimer's disease, like when is that happening, so we can apply interventions earlier, to better characterize dementia in Down syndrome, and also to aid in treatment development and the monitoring of new treatments. So I'm going to talk a little bit mostly about positron emission tomography. In our lab, we do some work with MRI. This is predominantly with Susan Buchheimer, and she does functional MRI and hippocampal unfolding work. but we're going to focus today on PET imaging, positron emission tomography. Now, historically in our lab, we've looked at glucose metabolism in the brain using what we call FDG, or fluorodeoxyglucose PET. And what we've done, and what others have done as well, is that in Alzheimer's disease, you have this signature pattern of hypometabolism, or low glucose utilization in the neurons in the brain. And for Alzheimer's disease, especially early on, you see a specific pattern of parietal, posterior cingulate hypometabolism. And this has been very useful for distinguishing among different dementias. For example, this is an Alzheimer's disease signature compared to a frontal temporal lobe dementia signature, which is, at least in the PIX form, or the frontal variant of FTD, you see hypometabolism in frontal regions of the brain. So this is a particular tool that's been used to help identify, not only diagnose Alzheimer's disease, or at least, let's just say, enhance the clinical diagnosis, but also use it for differential diagnosis and for early detection of Alzheimer's disease in people without intellectual impairment. Because we know that this pattern starts prior to the onset of dementia. You can see the beginnings of this pattern in people that have mild cognitive changes, so mild cognitive impairment, and also people with ApoE4 risk for Alzheimer's disease, and in people who are non-demented. Now, more recently, our group has been focusing on developing new probes, and I'm going to talk a little bit more about that today. One I'm going to focus on is called FDDNP, which is a plaque and tangle imaging probe. Now, there are other amyloid probes out there that are widely used, and Dr. Simmons talked about PIB, and this is also a very powerful tool for imaging amyloid deposition in the brain. So one way to think of the distinction between FDDNP and PIB is that FDDNP also images tau in the brain. Now, in persons with non-Down syndrome and people that don't have intellectual impairment, this is a kind of a classic schema of the development or the progression of neurofibrillary tangles and amyloid plaques in someone who's developing Alzheimer's disease. So Brock and Brock did autopsies on individuals that were of various ages, and they were able to essentially kind of map out how plaques and tangles, when they appear in the brain, and what the pattern is, because there appears to be a pattern of the deposition of these in Alzheimer's disease. And especially with the tangles, which seem to be a little more specific for the staging, in people without Down syndrome, you start early on, before even clinical signs manifest, before memory problems appear. You see some milder, smaller aggregations in the medial temporal regions. Now, as people become symptomatic, even in the early stages, the medial temporal region becomes very infested with tangles, and these tangles spread to other regions, more frontal and temporal regions of the brain. And eventually, when someone develops full AD, you can see that there is just an infestation of many, many regions of the brain. The amyloid has, at least if you read the literature, it's not as sensitive to staging of Alzheimer's disease. But again, in people without Down syndrome, it starts more in basal frontal anterior temporal regions, and then again spreads out as the disease progresses. So there are some differences, though, in this pattern between NFT and AP deposition in people with Alzheimer's disease without Down syndrome and people with Down syndrome. But one of the ideas, though, is if you're going to be developing a probe that images amyloid or amyloid and tau in the brain, for it to be considered viable or valid or useful, you would like to see some kind of correlation with the distribution or the uptake of your tracer in the brain, and it should map on to this type of amyloid and tau distribution that has been found in autopsy studies. So we have looked at the labeling or the ability of FDDNP to label plaques and tangles in brain slices and in living brains. And so here is a brain. This is a slice of brain. And FDDNP is fluorescent. So this green area is the FDDNP uptake in the cortex of the brain. Now, if we magnify this, so I can get this done, from this area of the brain, under fluorescent microscopy, you see that when you go a magnification of 400 times, what you see is an amyloid plaque and a neurofibrillary tangle. So we know that FDDNP is actually labeling these structures, pathological structures in the brain. We've also used it in humans, and what we found is that if you look at the global uptake of FDDNP in the brain, so that means if you look at a combination of regions of interest, like the total burden in the cortical regions of the brain, you see a complete separation, whereas people with Alzheimer's disease have the highest uptake. People in the transitional state between cognitively normal aging and Alzheimer's disease, right? This is called mild cognitive impairment. They're somewhere intermediate between people with what we believe are at least cognitively normal at the time of their imaging. So again, you see FDDNP lowest in cognitively normal individuals, highest in people in AD, and intermediate in people with MCI. This pretty much maps on to what we find in autopsy studies too. If you look at a specific region of the brain, what we found is that in the medial temporal region, which includes the hippocampus, that the uptake of FDDNP is very much at the level of Alzheimer's disease. So again, it maps on to the Brock and Brock staging in people without Down syndrome. And so these are actual images of individuals, representative individuals with normal cognition, MCI, and Alzheimer's disease over time. And so this is a control subject who remained a control subject over time. This is an MCI individual who converted to Alzheimer's disease, and this is the uptake of FDDNP in a brain of someone with Alzheimer's disease. You can see high deposition in medial temporal and lateral temporal and frontal regions. And to make the point is that the person with MCI who converted to clinically diagnosed Alzheimer's disease, you can see that there was an increase in amyloid and tau depositions, or I should say, increase in FDDNP binding over time. All right. And what we found is the person with at baseline who had Alzheimer's disease, we actually did an autopsy. This person willed their brain. And we found that the lateral temporal regions had predominantly amyloid and some tau. And that when we looked at the medial temporal region, it had predominantly tau and some amyloid depositions. And then this is a schematic of 8 controls, 6 people with mild cognitive impairment, and 6 people with Alzheimer's disease over time. So 20 people. What we found is that regardless of who you are, the worse your memory gets, the higher your FDDNP binding. So there's a correlation, right, of increased binding with worsening memory. Okay, and finally, we've also used this probe to look at ApoE4 carriers and non-carriers who are non-demented. And again, what you see in people who are having the ApoE4 risk for Alzheimer's disease and who are not demented, you still see higher FDDNP binding in these people than you do in somebody who's cognitively intact with no ApoE4. Okay, so now what we did was apply this to Down syndrome, and I think our previous speakers have made the argument of why Down syndrome is a great model for Alzheimer's disease and perhaps how these two areas can come together. So I'm not going to go into that, but instead tell you about what we did. Now briefly, we looked at subjects with Down syndrome who were non-demented, subjects with Alzheimer's disease, and cognitively normal subjects. And all of our subjects underwent FDDNP PET brain imaging. All right, and for those of you who work with Down syndrome, these are the DMR scales. So these are the scales of cognition and behavior in the persons with Down syndrome, okay? And these are essentially lower than what at least in the literature of people with dementia, these scores are lower. Pardon me? Okay. So what we found is that compared to normal controls, Down syndrome subjects showed significantly higher FDDNP binding in all brain regions. And so this is what this represents here. Compared to people with Alzheimer's disease, interesting finding, Down syndrome subjects showed significantly higher binding globally and in parietal and frontal regions. Okay. This is a representative of the parametric images of individuals, a control, a young Down syndrome subject, an older Down syndrome subject, and someone with Alzheimer's disease. And basically these, again, yellow areas represent higher FDDNP binding. This depicts there's a relationship between age and FDDNP uptake, okay? And also a relationship between dementia as well and FDDNP uptake. But you see that there's higher binding in the older Down syndrome than in the younger Down syndrome subject. And we found that, again, correlations between FDDNP binding in age were significant not only globally in the brain, but for a number of specific regions of interest. So the older the subject, the greater the FDDNP binding. And this showed that we did find some correlations. We're using non-parametric tests. This is a relatively small sample, and we are correcting this by age. But we looked at the neuropsychological, behavioral, and affect. That should be profile, not profile. And essentially, this is a scale that looks at 5 different domains of behavior in people with, it can be with not only Down syndrome, but head injury, etc. And it looks at inappropriateness, indifference, depression, mania, and language. And what we found is a correlation. Again, the more severe the inappropriateness behaviors as reported by collaterals, the greater the FDDNP binding. The greater the indifference in an individual, again, as reported by a collateral, the greater the FDDNP binding. And finally, I want to show that not only can we just use one probe, but we can look at a number of probes to assess Down syndrome and other dementias. Here we have our plaque and tangle imaging in an Alzheimer's disease, a person with MCI and the control. Here we have the FDG PET, which again shows neuronal circuit dysfunction, and which I didn't talk about, but I think could be very relevant, particularly because it involves hippocampal neuronal loss, is a probe called MPPF, which looks at 5-HT1A receptor densities, or serotonergic receptor densities in the hippocampus. And what you see is, again, a gradation of where you see higher plaque and tangle load or higher FDDNP binding. You see more neuronal circuit dysfunction as measured by FDG PET, and you see greater hippocampal neuronal loss. And that this is graded from most severe in Alzheimer's disease, in the middle in MCI, and of course, least affected in cognitively normal individuals. And we can't do this kind of work with just a lab or a person, and we've collaborated with many people at UCLA. We're also collaborating now with Dr. Lott, Dr. Tournay, and Eric is here, so Dr. Lott's group in Irvine, and to try to increase our sample size, and we have actually done some using the same measures to do this work. Oh, thank you.
Good. Questions then? Yes.
Postdoctoral studies here at University of Cambridge with Michelle Goeder, and my studies was focusing to study different compounds to use it for PET, and I saw that one compound called FSB was better than FDNP. My question is, do you know if this compound FSB can be used for your studies, or have you tried to use it? To use this compound?
We've used, right now we've used,
Thank you.
we've used both PIB and FDDNP, but we haven't used another compound yet, and MPPF. But we're, you know, we're open to using more or different compounds.
I saw that the FSB was better than FDN.
Do you know how it was better, what you mean by better?
FSB, the compound,
Yeah.
FSB, better than that you use, FDNP, because when you, my studies was just with slides, in vitro slides, but the fluorescence was better, and you can see more tangles and more amyloid plaques.
FDDNP does tend to have a lower signal. That's one of the weaknesses of it. So,
is there any correlation between your results and smelling loss in Alzheimer's disease?
We haven't looked at that yet, but that is a great question.
So, Linda, let me ask this question.
Yeah.
So, you do get a signal in using different methods pretty early. So the FDDNP study in Down syndrome showed something in a 21-year-old. Yeah. So my question, and presumably that's progressive over time.
Yes.
Do we have enough information to say how that progresses and whether or not that could be used in clinical trials to classify and define efficacy?
Right. No, and I think that is one of the gaps that we're talking about, that we don't have enough longitudinal data. We're seeing progression in our non-cognitively impaired group, but we haven't done enough in Down syndrome to know that, and that's why that is one of the...
How many people would we need? How many, in a prospective study, let's say over 10 years, how many patients would you need using the existing PET tools to begin to study the natural history of progression of binding?
Well, we've used about, I'd say about 60 of, AD and controls, so...
So maybe on that order.
Yeah.
Okay.
Can I add one more thing that I've
please.
learned today, and I thought it would be important to bring up, is that a number of our speakers have talked about depression and the onset of, of depression and Down syndrome. And one other parallel I think that's important to draw with Alzheimer's disease is you see a late onset depression that is a red flag for the future development of Alzheimer's disease in older adults. And so perhaps another piece of this puzzle would be, I mean, you're talking about the behavioral and psychiatric changes, are things like apathy and late onset depression, and how we can also compare Alzheimer's disease and Down syndrome in this venue as well.